Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Assalamu alaikum class, or I can say, or I should say, wa alaikum assalam class, especially for those students who have sent the message. It means at least few people have gone through the lectures. Alhamdulillah, that is good. Inshallah, more students are going to. In the last slide, these two structures were not discussed. These two structures, just I want to tell you that they have got the SO2 NH2 group. SO2 NH2 group. And in the SAR, one of the point is that we have also got the derivatives which have got the analgesic effect. These two compounds are the examples of analgesic compound, that is valdecoxib, celecoxib. They can be termed as selective COX-2 inhibitors. They have got the analgesic effect. They are different from the sulfonamide, but as they have got the SO2 NH2, that's why they are also included in the class of sulfonamide, not the sulfonamide, but sulfonamide compounds. Sulfonamide compounds. So this slide only is used to show the structure of the analgesic compounds, or the compounds having the SO2 NH2 group. May be structured different from that sulfonamide. One more thing, when we have got the structurally different compounds, we can also observe that the mode of action is going to be changed. The mode of action is going to be changed. Here we are saying that the different derivatives have got different effects. The antibacterial, the anti-diabetic, diuretic, analgesic. Okay. But when we are going to study the mechanism of action of sulfonamide, then we are using another term. What is that term? What is that term? The true sulfonamide and the false sulfonamide. The mechanism of action which we are going to discuss: if any molecule is fulfilling that criteria, then they will be termed as the true sulfonamide compound. Otherwise, the compound maybe have got the activity the other activity may be termed as sulfonamides, but they are not true sulfonamides. In this slide, you have seen the word sulfonamide, but it is better to use the term sulfonylamide here, or derivatives of sulfonylamide, or derivatives of sulfonamides. Now I have told you and I have asked you to go through those terms, the antibacterial compound, why we are not saying that it is antibiotic, why we are saying bacteriostatic. You know, by definition, antibiotic are the compounds which are obtained from a living system, from a living organism, and then it is used against some other living organisms, some other living system in a minute doses. Those compounds are termed as antibiotic. And when you are using the term antibiotic, what is anti means? They are against that one, and they are obtained from a living system, but they are used against the life or against those living system, maybe the bacteria, maybe the protozoa, maybe the other compounds. But when we are using the bacteriostatic, antibacterial, it means we are talking about the bacteria, antibacterial compounds. Sulfonamides. In the first lecture, in the introduction, we have seen that sulfonamides or sulfonylamide is obtained from a red dye that is prone to seal, and the different derivatives are synthesized from that one, sulfonylamide. What does it mean? All of these compounds are the synthetic compounds. They are obtained from the red dye or maybe from other sources, but not from the living system. And when they are not obtained from the living system, they cannot be termed as antibiotic, although that classical definition is somewhat modified. That modification says that the molecules, if they are completely synthesized in a lab, or we have got the semi-synthetic derivative, but initially that molecule was obtained from a living system, even then that synthesized molecule can be termed as antibiotic. We have seen that a number of antibiotics, the penicillin derivatives, cephalosporin derivatives, and the antibiotics, now we are getting those compounds from the lab as a synthetic molecule, but initially all of those were obtained from some living source. That's why they are termed as antibiotic. And here for sulfonamide, generally or in most of the cases, we are not using the term antibiotic. Although I have seen few books, I can't say why they have used that term, but that term was used. I don't know why. I have no justification for that one. Now we can say that sulfonamide are the antibacterial compound, and antibacterial compound may be bacteriostatic or they may be bactericidal. Bacteriostatic means they are going to inhibit the growth of the molecule, the growth of the organism, and bactericidal, they are going to kill the organism. Sulfonamides are used as bacteriostatic, can be used or can become bactericidal, but for that purpose the dose should be increased, but that dose will be toxic. That's why at that dose we are not using the compound, and hence the compound have not got the bactericidal action. Instead of that, we are using sulfonamides as bacteriostatic agent. Another term here used is the antimetabolite. What does it mean by antimetabolite? When we are using the term antimetabolite, we are saying that any compound which is structurally related to some metabolites in a specific metabolic pathway, due to the structural similarity, that molecule can enter into the specific metabolic pathway, but as it does not have got the same or 100% similar structure, have got some differences in the structure. That's why that compound cannot serve the purpose of that metabolite, and hence we are using the term antimetabolite. Simply we can say that antimetabolites are the compounds which have got certain structural similarity to the metabolite of a specific metabolic pathway, can enter into the metabolic pathway, but due to the differences in the structure, that compound cannot serve the purpose and termed as antimetabolite. And what does it mean? Instead of participating in that metabolic pathway, rather is going to inhibit that one. It is going to inhibit that one. They are maybe going to inhibit the metabolic pathway, or the resulted product of that metabolic pathway will be different from the required product of that metabolic pathway. Just we have talked that antimetabolites are those compounds which can enter the pathway. Here PAPA is one of the compound, that is para amino benzoic acid. You can see the compound have got almost the same structure. Only one functional group, that is SO2 NH2, is replaced by COOH. Is replaced by COOH. It means the sulfonamide have got the similarity with the structure. This moiety or up to this moiety we have got the same structure, and then only the difference is in the second functional group, or SO2NH2 is replaced by COOH. Now this PABA is required for the synthesis of folic acid. Just I am very briefly give that mechanism. Pteridine with PABA going to synthesize dihydrofolic acid. The enzyme is dihydrofolate synthetase. Then this dihydrofolic acid will be converted into tetrahydrofolic acid and then to folate cofactors. Those folate cofactors act as one carbon donor in the synthesis of different nucleic acids, which are required for the normal growth of the cell. When these compounds, the sulfonamide or its derivatives are used, due to this structural similarity, they can enter into the pathway of that synthesis of folic acid, but they have got not the same structure. That's why the product will not be there. PABA compete for the enzyme that is dihydrofolate synthetase. PABA compete with the enzyme that is dihydrofolate synthetase, or we can say that PABA or sulfonamide is competitive inhibitor of PABA. Sulfonamide is competitive inhibitor of para-aminobenzoic acid or PABA. They compete for the same enzyme, that is dihydrofolate synthetase, and hence due to differences cannot serve as a substrate for that enzyme, and the further pathway is blocked. Dihydrofolate is not produced. Tetrahydrofolate will be inhibited, and hence folic acid cofactor, folate cofactors are not there, or the synthesis of nucleic acid is not possible or is interfered, and the growth and the maintenance of the normal cell is inhibited, or we can say that the action is the bacteriostatic. They are going to inhibit the growth of the cell, because for that the bacteria required the folic acid or folate cofactor. As I have told you that folate cofactor will serve as one carbon donor in the synthesis of nucleic acid. Hence, this mechanism is termed as the competitive inhibition, and the molecule or the drug, the compound, is termed as anti-metabolite. So the sulfonamide or its derivatives are the competitive inhibitor of PABA. They are anti-metabolite. They have got the bacteriostatic activity. They have got the bacteriostatic activity. Then we have got another point which should be discussed, and I have told you that I've got different concepts. Here we have got another concept that is termed as concept of selectivity. You know, folic acid is required by the bacterial cell, but also folic acid is required by the mammalian cells or for the mammalians like human beings. Then how can we use those sulfonamide? It means the folic acid is also going to be reduced in the mammalian with the bacterial cells. May be possible to very less extent. Why? Because we have got the selectivity of the molecule against the bacteria. Or in other words, we can also say that mammalian can use it selectively against the bacteria. And how we have got the selectivity, or how the molecule achieves the selectivity? We have got at least 2 points of differentiation. One is bacteria has a susceptible enzyme which is not present in mammalian cell, Second, bacteria lacks the transport protein which would allow it to acquire folic acid from outside the cell. What does it mean? It means that if the folic acid is present in the medium of bacteria, even then bacteria cannot use it because the transport protein is not there. While in the case of mammalian cell, the transport protein is present, and hence we can use the folic acid from outside the cell, while mostly it is not synthesized due to lack of that susceptible enzyme. Next, we are going to discuss the toxicity of the molecule or the side effect of the sulfonamide or sulfonamide, toxic and side effect of the sulfonamide. We have got a number of side effects, including crystalluria, or crystalluria can be termed as the main toxic effect, while the allergic reactions or this type of the reactions can be considered as the side effect, can be considered as the side effect. Before dealing with that one, the few terms like pH, pK must be defined. pH, a scale used to specify the acidity or basicity of an aqueous solution. pK is one method which is used to indicate the strength of an acid. pK is the negative log of the acid dissociation constant or Ka value. A lower pK value indicates a stronger acid. The lower pK value indicate a stronger acid, and we are going to consider these definitions mainly with the resolution of that problem, that is the problem of crystalluria again. We have another term or new term, that is crystalluria. Crystalluria is the formation of crystals in the urine. Mind it, when we are urea, URIA, in the word, that indicate something in the urine. For example, crystalluria, crystals in the urine. Glycosuria, glucose in the urine. Proteinuria, protein in the urine. Hematuria, blood in the urine. And when we have used EMI, emia, that indicate something related to the blood. Glycemia, glucose in the blood. Proteinemia, protein in the blood. Septicemia, sepsis in the blood. Anemia, what does it mean then? Anemia. In blood we have got the hemoglobin, and when anemia means without hemoglobin, although it is not without hemoglobin, but the level of hemoglobin is less in the blood, or less hemoglobin in the blood, the condition is termed as anemia, and without hemoglobin or without blood. So that should be noted, crystalluria. Now I have told you that crystalluria, or formation of crystals in the urine, is the main toxicity of the molecule. The compounds sulfonamides are used for the infective conditions. Say in the UTI, urinary tract infections, we are using sulfonamides for the treatment. But if the molecule result in the formation of crystals in the urine. It means the patient is going to suffer from another problem. That's why that is a toxic effect of the molecules. Now you know that when we start the medicinal chemistry, generally we start with the SAR and the effect of the different parameters on the SAR or biological activity, physical parameters such as temperature, pH, pK. Those initially studied terms are used now here or applied here to resolve the problem of crystalluria. First or initially, we must know why the crystals are formed or how the crystals are formed. If we are able to know the mechanism by which the crystals are formed, then it will be easy for us to resolve or solve the problem. Here the pH and pK will play an important role in the formation of crystals. Inshallah, we are going to deal it in the next lecture, that is about the toxicity of the sulfonamides. Inshallah.